Shri Gurubhyo Namaha children. I am happy to welcome you all for our online teaching academy, Ekalaiva. For the class 12, subject BioBotany, chapter 5, plant tissue culture. Today, we are going to discuss about techniques involved in plant tissue culture. Before going to see the topic in detail, first we will see the basic steps in plant tissue culture. See the picture here, children. What is this? A plant. This is said to be mother plant. A small portion of plant, that is leaf. We have to separate the leaf from the mother plant, and the small portion is said to be X-plant. X-plant is nothing but a small segment or small portion of the mother plant is said to be X-plant, and we take a small portion. In that we just separate the tissue, and this tissue is cultured in a culture medium, that is nutrient medium or growth medium. That growth medium have all the essential nutrients for the tissue to grow. Okay. After that, the callus will develop. Callus is nothing but a tissue. Is the undifferentiated mass of cell. Undifferentiated callus is formed. After that, the callus is separated from here and it is cultured again in a conical flask. The callus separated and a single cell, small one cell is cultured. After that, further a new plantlet will develop. This is the basic steps in plant tissue culture. Now we will see the technique involved in plant tissue culture. These are the steps. The first one is sterilization, second one is media preparation, third one culture condition, fourth one induction of callus, fifth one embryogenesis, and the last one is hardening. These are all the techniques, these are all the steps, or these are all the procedures involved in plant tissue culture. What is the first one I said? Sterilization. What is the meaning of sterilization, children? Cleaning. Cleaning, or that is free from pathogens, free from microorganisms, that is sterilization. Second step, second one is media preparation. Media preparation is very important for plant tissue culture. If there is no proper media preparation, means the plant will not grow. What is the basic concept in plant tissue culture? From the small X-plant, we can able to produce thousands of plantlets. That is a basic concept in plant tissue culture. You already heard about the totipotency, differentiation, redifferentiation, etc. Right? And third one is culture condition. So we have to maintain the proper pH, proper temperature, etc. Fourth one, induction of callus. We just induce the tissues to form a callus, right? And then formation of embryo, that is called embryogenesis, and the last one is hardening. These are all the basic steps or techniques involved in plant tissue culture. Now we will see the first one in detail. The first one is sterilization. As I said earlier, sterilization is nothing but it is a technique or it is a method employed to get rid of microbes, that is free from microorganisms such as bacteria, fungi in a culture medium as well as vessels and in explants. We have to remove we have to remove, that sterilization is a technique to remove or destroy the microorganisms which is present in the culture medium, vessels as well as in the explant. Then maintenance of aseptic environment. Aseptic environment is nothing but the procedure followed by removal of pathogens. Okay, the procedure followed by removal of pathogens. We have to follow strict rules and regulation from pathogens, infection of any organisms. So during in vitro culture, in vitro means the culture techniques takes place in the laboratory. During in vitro tissue culture, maintenance of aseptic environment condition should be followed. That is sterilization of glasswares, sterilization of forceps, then sterilization of other equipments, other apparatus by autoclave at 121 degrees Celsius for about 15 to 30 minutes, or dipping the apparatus or dipping the accessories in 70% ethanol, followed by flaming and cooling. This is the first step in sterilization, maintenance of aseptic environment. Clear, children? The second step in sterilization is sterilization of culture medium or culture room. Okay. So floor and the walls of the culture room, first we have to wash with detergents, and then followed by 2% sodium hypochlorite or 95% ethanol. So this sodium hypochlorite or ethanol eradicates, destroy all the microorganisms which is present in the culture room. See the children, see the picture, children. What is this? Have you seen before this? No. What is the name of the apparatus? It is an instrument, right? What is the name of the instrument here? This is a laminar air flow. Is it? See, the cabinet of laminar air flow, this is a cabinet, full cabinet, is sterilized by cleaning the work surface. This is a work surface. Cleaning the work surface with 95% ethanol, and then it has to expose of UV radiation, ultraviolet radiation for 15 minutes. First we have to clean the workstation with 95% of ethanol, followed by the exposure of ultraviolet radiation for 15 minutes. Then this laminar air flow is sterilized. Okay. The third sterilization, sterilization of nutrient medium or growth medium. Okay. So culture media or growth media is dispersed in none. That means that growth media, culture media is poured in a glass containers, and it is plugged, it is plugged with non-absorbent cotton or sealed with plastic closure or any aluminum foil. After that we have to sterilize by using autoclave of temperature 121 degrees Celsius for 15 to 30 minutes. See the picture here, children. This is the autoclave. Autoclave is an instrument used for sterilization, right? So after dispensed the culture medium in the conical flask, and it is sealed with, it is covered with aluminum foil or cotton plug. After that we have to sterilize the nutrient medium. Now the nutrient medium is there in the conical flask. This instrument, this apparatus containing the nutrient medium has to sterilized. We have to keep this in the autoclave for about 15 minutes at temperature 121 degrees Celsius. After that the culture medium gets sterilized. The removal of this particular medium is free from microorganisms. Okay. Then fourth step in sterilization, sterilization of explants. The last, that is explant. Explant, you know very well, a small portion. Explant is nothing but a portion of plant parts, that is root, stem, leaf, etc. Okay. So finally we have to sterilize the explant also. Initially the plant materials to be used for tissue culture, whatever it may be a root or leaf, anything. So the plant material to be used for sterilization or used for tissue culture should be surface sterilized. That means first exposing the plant material in running water, and then it is treated it in surface sterilization agents like 0.1% mercury chloride and then 70% ethanol under aseptic conditions. Aseptic conditions is nothing but free from microorganisms inside the laminar airflow. Initially the explant has to be surface sterilization. It has to be by exposing the material in the tap water, running tap water, followed by the treating, followed by the explant, followed by treating the explant in the surface sterilization agents like mercury chloride and then 70% ethanol under aseptic condition inside the laminar airflow. See the picture here, children. Now this is the small part of your plant. It is explant. This explant is surface sterilized by exposing the material in the tap water, running tap water. Clear, children? First we have to clean the surface by introducing this explant in a running tap water. So all the dirty is exposed, then followed by the sterilization agent, that is 1% mercury, 0.1% mercury chloride and 70% ethanol under aseptic conditions inside the laminar airflow. Okay. Then second step in the this is media preparation. Second technique in plant tissue culture is media preparation. The success of plant tissue culture lies on the basis lies on the composition of medium what we are taking, right? So the main success is based on the composition of growth medium, plant growth regulators and culture conditions such as temperature, pH, light and humidity. If anything is damaged, if anything is not at all at optimum level, means the plant will not grow nicely. Okay. So we have to take serious steps, we have to follow the media preparation procedures properly. Okay. Suitable nutrient medium, suitable nutrition medium as per the principle of tissue culture preparation and we have to follow. MS medium means Morashik and Snoog in the year 1962 it is discovered, is commonly used as a nutrient medium. It has carbon sources and then which is suitable and having suitable vitamins and hormones. So usually we are using the MS medium for preparation of media in the tissue culture. See the picture here, children, the conical flask containing culture media. So a medium may be solid or liquid. For solidification we have to add a gelatinous agent called agar. Agar is a solidifying agent. It is extracted from marine algae. Clear, children? So if it is liquid means we have to add agar on that medium, then it became solid. The solidifying agent is agar, it is extracted from the marine algae. Then third technique is culture condition. First one is pH. The pH of medium is normally adjust between 5.6 to 6 for the best result. So 5.6 to 6 means it is acidic in nature, below 7 it is acidic only. Then temperature, The culture should be incubated normally at the constant temperature of about 25 degree Celsius plus or minus 2 degree Celsius for the optimum growth. Clear, children? Then humidity and light intensity. The culture requires 50% or 50 to 60% relative humidity at 16 hours of photo period by illumination of cool white fluorescent tubes. See here, this is the lab, this is a culture, plant growth culture or plant tissue culture having the light intensity of about 1000 lux. Okay. And then aeration. Aeration is to the culture can be provided by shaking the flask or tube of liquid culture on automatic shaker. See the picture here, children. This is the automatic shaker. We have to keep this flask containing liquid medium, and using this instrument we just shaking all the flask. So aeration takes place. Aeration to the culture can be provided by shaking the flask or tube containing liquid culture on the automatic shaker. This is automatic shaker, okay, or aeration of the medium by passing with filter sterilized air. By passing the filter sterilized air, now the liquid medium get aerated. Clear, children? Then induction of callus. As I told yesterday itself, callus is nothing but the undifferentiated mass of cell, right? So see, explant of about 1 to 2 centimeter. See the size, very small part of your plant. 1 to 2 centimeter of sterile segment of selected from the leaf, from the plant. So explant of about 1 to 2 centimeter selected from leaf, stem, tubes, tuber or root, whatever it may be, and it has to be inoculated in the MS nutrient medium. What is inoculation? Just transferring the explant to the sterile glass tube, sterilized glass tube containing nutrient medium, that is called inoculation. Initially the explant, the sterile explant, that is 1 to 2 centimeter selected from leaf, root or stem is inoculated. That means transfer of explant to the sterile glass tube containing nutrient medium, particularly MS medium, and then supplemented with some plant growth hormones, that is auxins, supplemented with auxins and incubated at about 25 degrees Celsius plus or minus 2 degrees Celsius in an alternate light and dark. Alternate light means light and night, light dark. Alternate light and dark period of about 12 hours to induce cell division. After that, as soon as the upper surface of the explant develops as a callus. So after the incubation period is over, the upper surface of the explants give rise to callus. Callus is nothing but the undifferentiated mass of cell. See the picture here, children. The upper surface of the explant develops as a callus. See, this is the organogenic callus and this is embryogenic callus. Callus is a mass of unorganized growth of plant cells. Unorganized are nothing but undifferentiated. So unorganized or undifferentiated, both are same only. Callus is a mass of unorganized growth of plant cells or tissue in vitro condition, in vitro culture medium. Clear? Then fifth step, that is fifth technique, this embryogenesis. Wherever you heard the word genesis means formation, embryogenesis means formation of embryo. Okay, the callus cells, we already discussed about the induction of callus. Now we have callus. The callus cells undergoes differentiation and produce somatic embryos. They are said to be embryoids. The somatic embryos are called as embryoids. The embryoids are again subculture to form a plantlet, to produce a plantlet. See the picture, the test tube containing plantlet. Okay, the callus undergoes differentiation to form a embryos. The embryos are called as known as embryoids. The embryoids are subcultured. Again, it has to be cultured to form a plantlet. Now the test tube containing plantlet. Clear? Then sixth one, last step, hardening. The plantlets develop in vitro conditions. They need hardening period and so are transferred to greenhouse or hardening chamber and then to a normal environment conditions. So initially the plants require the hardening chamber, or that is for the greenhouse place. Okay. So hardening is the gradual exposure of in vitro plant developed in the humid chamber is diffused light for acclimatization so as to enable them to grow under normal field conditions. So initially hardening is nothing but the gradual exposure. So previously all the plantlets are grow only in the in vitro condition, it is in the laboratory only. The plants require the normal environment. So hardening is a gradual exposure of in vitro developed plantlets in humid chamber in diffused light for acclimatization so as to enable them to grow in a normal condition, normal environmental field conditions. Clear? See the picture here. This is the humid chamber. All the plantlets are in the humid chamber. This is called hardening. After that they are ready to the normal environment. Okay. Thank you children. We will see in the next class.